recording to the... Okay, so this meeting will be recorded. We muted your microphones in order to leave only chat option for some questions. We would kindly ask you to close your cameras in order to keep your anonymity. We will give the chat at your disposal throughout the workshop duration, and we kindly ask you to actively participate, as not only at the end of this workshop we will have space for Q&A questions, but also we left some time after each presenter that you can ask the question, as maybe some of you won't be able to join us till the end of the workshop. We are starting with a short introduction of both projects. So Dr. Janja Vidmar will give presentation of InPlasTwin project as a project coordinator, and Piaton is a teaming project manager, and she will give presentation of VITO project. After the short presentation of the projects, we are continuing with our speakers. We have 4 speakers today. First presentation will be given by Dr. Phan on microplastic analysis in soil, from sample preparation to quality assurance and quality control, which will be followed by a presentation of Dr. Fred Olav Libnau from Institute of Marine Research on separation of microplastics from marine sediments. After that, we envisage short coffee break, just to refresh ourselves and prepare for the second part of the workshop, which will start at 11:30 with the presentation of Dr. Sevil Wafadar-Afshar on Olav extraction of biodegradable microplastic in compost. And this presentation will be followed with a presentation of Dr. Eleni Christoforou from VITO project on tracking microplastic in Biota, hands-on sampling and extraction techniques. At the end, we have left half an hour for a Q&A section and to brainstorm actually this and tackle the main questions that arise regarding this microplastic hot topic. Before we start with the presentation, I will shortly introduce our speakers. Dr. Phan is a postdoctoral researcher at VITO Institute, focusing on developing methods to analyze micro and nanoplastic in complex environmental samples. She earned her PhD as Marie Curie ITN Fellow at Lancaster University, UK, where her thesis was titled Micro and Nanoplastic in Soil, Analytical Methods and Environmental Fate. At the moment, she contributes to Horizon Europe projects on micro and nanoplastic investigations, including InPlasTwin and INSPIRE project. Now I'm presenting Dr. Sevil Wafadar-Afshar, who comes from a technical university of Denmark. She is a postdoctoral researcher at the department called DTU Sustain, and her research is focused on the environmental fate and effects of bioplastic. She obtained her PhD from DTU Sustain with the thesis Fate and Effects of Bioplastic in Waste Management System and the Environment. And throughout her PhD, she examined degradation behavior, microplastic formation, and ecotoxicity. She currently contributes to the Horizon Europe project InPlasTwin, investigating the environmental fate and effects of biodegradable mulching films. Dr. Fred Olav Libnau is a presenter from Institute of Marine Research, Norway, where she actually earned her PhD at the University of Bergen, Department of Chemistry. Previously, he has experience as he worked in pharmaceutical and R&D and production facilities with in-on at line analysis, production, process development, and optimization. For the last 4 years, he has been working at Institute of Marine Research with focus on density separation of microplastics from marine sediments. And Dr. Eleni Christoforou is an associate scientist at the Cyprus Marine and Maritime Institute, participating in European and national projects with such as PUREE-Y, PLASTIER, and EDNL-EV, researching topics like climate change, marine pollution, and local biodiversity. She holds a bachelor degree in marine biology and a BA in art from the University of California in Santa Cruz, while she earned her PhD in marine biology from University of Glasgow. Her doctoral research focused on the effects of microplastic and light pollution on the ecosystem services provided by blue mussels. Thank you for this short introduction which I gave. And now I give floor to project coordinator of InPlasTwin project, Dr. Janja Vidmar, who will now share her presentation.
Good morning, everyone, also from my side. And I would like to welcome you to the first joint online workshop of the PUREE-Y and InPlasTwin project. As Gordana already told, I'm Janja Vidmar from the Jožef Stefan Institute, and I'm very happy that I have the opportunity on today's event to give a short overview of the InPlasTwin project. So the InPlasTwin is a project funded under the Horizon Europe program and brings together 6 different partners from 6 countries to increase our expertise in micro and nanoplastics analysis through a twinning approach. Coordinated by the Jožef Stefan Institute and running from October 24 to September 27, this project aims to address one of the most pressing environmental issues nowadays. This is the pollution with micro and nanoplastic. The primary objective of the InPlasTwin project is to increase the research and innovation capacity of the Jožef Stefan Institute from Slovenia and Agricultural University of Athens from Greece in the field of micro and nanoplastics analysis. This will be achieved by networking, knowledge sharing, and research activities with leading research institutions in Europe, with a long-term aim to deepen our understanding of environmental and food-related impacts of micro and nanoplastics, with a particular focus on agriculture. So this goal will be achieved through several specific objectives. First, by fostering long-term collaboration among project partners and sharing the knowledge in studying the micro and nanoplastics in environmental and food samples. The project also aims to enhance partners' scientific and innovation capabilities, also giving them increasing their expertise in micro and nanoplastics analysis. At the same time, InPlasTwin project also aims to improve the project management skills of the coordinating institutions and promotes diversity, inclusion, and ethical practices in the research. And finally, by increasing social impact, the project not only adds the visibility and improves the visibility of the project partners, but also raise awareness of the micro and nanoplastic pollution among various target groups. So the InPlasTwin consortium consists of 6 partners covering very different fields of natural sciences, from the environmental analytical chemistry to agriculture to environmental engineering, and also innovation management. The Jožef Stefan Institute and Agricultural University of Athens, both from the widening countries, are in the InPlasTwin project focus on improving their skills and knowledge on the micro and nanoplastics analysis, with AUA also contributing their extensive knowledge on the greenhouse crop cultivation. Then we have 3 next partners who are experts in micro and nanoplastics analysis, including Flemish Institute for Technological Research from Belgium, Technical University of Denmark from Denmark, and Institute of Marine Research from Norway, who will support capacity building activities in the InPlasTwin project by providing their knowledge and hands-on experience to other project partners. And Foodscale Hub from Serbia is leading the dissemination, exploitation, and communication activities to improve the social impact of the project and its results. Since this is a twinning project, of course, the main activities are focused around the building partners' capacity in analyzing micro and nanoplastics in environmental and food samples. This will be done through a series of different laboratory experiments combined with the advanced analytical techniques. First, we are studying the degradation of agricultural plastics, in particular mulching films, under the realistic agricultural conditions, also with the aim to study the release of micro and nanoplastics from this experiment. Next, we are also getting training in different sample preparation protocols for extracting micro and nanoplastics from different environmental and food samples. Thirdly, we are gaining our expertise in different analytical techniques that are used for the quantification, identification, and characterization of micro and nanoplastics. And last, we are involved also in the investigation of plastic, organic, and inorganic additives that are present in mulching films and their release into different environmental and food samples. So all those capacity building activities revolve around a case study on strawberries, where we are investigating the uptake and impact of micro and nanoplastics that are being released from the mulching film degradation. These films are commonly being used in agriculture for the crop cultivation, including strawberries, because they increase the crop yield. However, their improper disposal or management can lead into the accumulation of plastics in the soil, which can then break down to smaller micro and nanoplastics. And these small particles can persist in the soil, affecting plant health and also potentially entering the food chain. And given the very limited research in this area, we decided that this case study will be a core of the project research component. So all the knowledge that we are gaining through the capacity building activities will be at the end, in the second half of the project, applied and tested for the analysis of micro and nanoplastics and also leachable plastic additives in strawberries collected from two different types of experiments. In the first experiment, hydroponically grown strawberries will be exposed to micro and nanoplastics released from the degradation of mulching film. And in the second experiment, a small-scale field experiment will be conducted in the commercial farm in the Western Peloponnese in Greece, where the strawberries are being grown with the use of different mulching films. So in these two experiments, we are not only going to study the potential uptake of micro and nanoplastics and plastic additives by strawberries, but we will also evaluate the impact of those plastics on the strawberry yield and quality attributes of the strawberry fruit. At the end, I would just like to highlight the project progress and the main activities that we conducted in the first year of the project implementation. We have been mainly focused on performing training activities at the partners' institutions, building expertise in different types of activities. Our project and its results has been presented on several occasions and events, conferences, symposium, reaching not only the scientific community, but also strawberry growers and also the general public. Our project has been very actively promoted on the project website and social media channels. And of course, we are also building synergies with sister projects that are also focused on microplastic research. And today's workshop is a good example of such collaboration with the PUREEF-Y project. So I wish all the participants to enjoy in today's workshop. I hope that you get some useful insights into different approaches that our presenters will talk about regarding the sampling and extraction protocols for microplastics in complex samples. I wish you successful and enjoyable event, and thank you for your attention.
Dear Janja, Thank you so much for giving introduction of on InPlasTwin project, and accordingly we are giving a word to Gotelin Piaton, who is project coordinator of PUREEF-Y project.
Thank you, Gordana. Thank you, Yanya, and good morning to everyone. It's my pleasure to be with you this morning, and I hope that you can start seeing now the presentation about the PUREEF-Y project. But before giving you more information about this twinning project, I would like to really extend warm thanks to the whole team of the InPlasTwin project, of course, under the coordination of Yanya, for the opportunity to co-organize and co-host this webinar today. I am really impressed by the number of participants, and that actually confirms the very high interest in the topics that will be discussed today. So in my turn, thank you for this and welcome to all participants. A few words about PUREEF-Y. PUREEF-Y stands for Past, Present, and Future Status and Mitigation for Cyprus Shallow Water Reef Biodiversity and Functioning Under Global Change. We are actually also a twinning project, but obviously we focus on different types of environment as being a marine and maritime institute. So in our project, a few key facts about the project. It's a very similar duration, 3 years. We have started in July 2024, and we have a smaller consortium of 3 partners coordinated by CMMI. We have the chance to work with the Israel Oceanographic and Limnological Research Institute in Israel and the Autonomous University of Barcelona. Overall, the objective, building on the complementarity of expertise of our advanced partner, is to enhance the progress of CMMI as a 6-year-old research center in marine and maritime research, innovation, and technology development towards scientific leadership in the fields of chemical oceanography, marine ecology, as well as experimental design and marine pollution, and of course to be able increasingly to exercise influence when it comes to management plans for the marine and coastal ecosystem and environments in the Eastern Mediterranean, in the Mediterranean overall, in Europe, and why not even globally if we raise, of course, even our ambition. So you can see here a few pictures. It's indeed a capacity building and knowledge transfer type of project. We spend a lot of time on the surface or inside the water, indeed, going sampling, going collecting data, but also a lot of time actually in our labs, getting trained, transferring knowledge from our advanced partners. The common point in all of those activities right now, 15 months in the project, is the big smile that you can see on everybody's faces because we are engaged in doing something that we are truly passionate about. Key components of PUREEF-Y as a project are the fact that we have coordination and support activities. We established together a scientific and innovation roadmap. We organize a lot of training and capacity building within the consortium, but also with external partners to increase our scientific skills, to improve our research capacity. We also undertake training and capacity building related to innovation management, project management, even entrepreneurship. And of course, we try to relate the research that we perform with any kind of potential impact we can have on research and innovation policymaking, but also policymaking for the protection, restoration of the oceans and the waters overall. The project has also a research and innovation component project within the project that I will present into more detail in a couple of seconds. But it has also networking activities during which we will have the chance to, of course, have joint publications, scientific publications, but things that can interest our participants today. A summer school that will take place in Cyprus. So we'll see whether spring is more appropriate though because of the high temperature. researchers we sometimes experience. We organize webinars and seminars, in particular for early career stage researchers, and you'll all have the chance possibly to join our scientific conference that will happen in May 2026 next year. We have the pleasure to network and to establish collaboration, for example, with InPlasTwin partners and many others. So with regards to our research and innovation project, we have 4 pillars in this research and innovation project. The first pillar is really to collect data, to sample, and to be able to track physico-chemical parameters related to climate change and to ocean acidification in the Eastern Mediterranean. The idea is to be able to possibly extract information to better understand and predict the effects of climate change and ocean acidification in the future. We know it might be accelerating, but we need to understand better the effects. In a second pillar, we also perform an ecological assessment of benthic marine biota to assess the current state of intertidal and shallow subtidal macrophyte communities, biodiversity, and the health of these ecosystems. So I'm really pleased that today one of our scientists, Dr. Eleni Christoforou, has the chance to highlight the work that she performs in the project. In a third pillar, we sample a lot. Actually, every 3-4 months, we perform seasonal sampling in our waters. We sample coastal waters, but we also sample sediments in order to possibly identify marine pollutants at key locations around Cyprus. And obviously, nanoplastics and microplastics are within our radar, not literally speaking, but indeed, this is something that we aim at being able to detect. And of course, we want to understand the influence on anthropogenic activities with regards to their presence and to assess pollutant accumulation over time. And then in a fourth pillar of our research and innovation component, we want to test the functioning of benthic communities at different ecological states in Cyprus, to obviously use methods, including mesocosm experiments, that are actually transferred to us by our colleagues from the IOLR Institute. So, conclusion, I would like to highlight that indeed very active projects. You're all invited, if you are interested in the type of research we do, to our project conference that is dedicated fully to marine sciences. It will take place on the 21st and 22nd of May 2026 in Larnaca, Cyprus. So feel free to scan the QR code to get to know more about the event, and feel free, of course, to follow and to share what we do in these projects. It's really our pleasure to be here today and to share widely the good work that is being performed in the twinning projects and InPlasTwin and PUREEF-Y in particular. So wishing you a very successful webinar, and thank you very much once again.
Thank you very much, Gotalin. I'm very pleased that we have this project together and that we recognize collaborations that can be undertaken starting from this workshop. And as this is also, we are starting our second year, you are in the first year of the project. I'm sure that throughout the project's lifetime, we will organize more events together. To start officially with the workshop and with presentations of scientific part, I'm calling now Dr. Phan to share her screen with us and give us her insights into sampling and microplastics from soil as the first matrices that we will cover today during our workshop.
Thanks, Gordana. Thank you. I'm sharing my screen now.
Thank you. Yes.
Can you all see my screen?
Yes. Just you can put it in the presenter. Oh, full screen.
So,
Yes.
hi everyone. Thank for joining the workshop today, and it's my pleasure to share with you some insight from microplastic analysis in soil, from sampling to sample preparation, and also some QA/QC consideration. And I'm from VITO, in Belgium. So microplastic is now present everywhere and is also in our soil. And it's really important for us to know how much plastic are really present in our soil. However, this is not an easy task, and especially being able to reliably quantify microplastic in soil is really tricky, and is from soil sampling to really extract microplastic from soil and then identifying and quantifying microplastic in soil, we can bring error and uncertainty in every of these big stuff. And it's really important to really minimize the error and so that we can be sure what we are measuring. So I will first talk about sampling of microplastic in soil. So when we consider sampling soil for microplastic, there are several things that we need to consider. Which sampling approach should we take? How much area and what is the volume of soil we should take to have representative soil sample? And then how deep should we sample the soil? How often? And also what type of equipment should be used for soil sampling? And of course, these should be depend on the research questions and on also the economic concern. So first about sampling approach, there are six type sampling approach that we could have, like judgmental, which is normally used when we have prior knowledge of our field. For example, we know the historical application of plastic, or we see that some of the field are contaminated and we know some field are not contaminated. And then also simple random sampling that is normally used when we have a uniformly contaminated field and we want to randomly sample the soil so that we will avoid unbiased statistical analysis. And then we have systematic grid sampling, meaning that we divide our field into several grids and then we take the different points. And this is especially useful when we want to delineate the hotspot within a field, or we want to study the gradient of the plastic contamination in a field. And next one, we have transect sampling. This is also useful when we want to study how microplastic contamination spread along linear feature. For example, we want to study how the tire wear particle contaminated along the road, and this is a useful approach for that. And then we have unaligned grid sampling, which is kind of a mix between simple random sampling and systematic grid, where we divide our field into this grid, but then we take randomly the point within the grid. This allows us to spread along like throughout the field, but also allow to do random sampling to avoid unbiased and statistical analysis. And then next one, we have stratified sampling. We divide our field into several area, but then we also take sub-sample in this area to see the variation within those sub-samples. So for microplastic analysis in soil, there is a literature review studying 106 study, and they highlight that most of the study use random sampling. In our case, in our lab, we also mainly use random sampling, and then some use transect and then grid sampling. The next thing we consider is the type of equipment. So of course, we are sampling microplastic. It would be useful to avoid plastic equipment. So a lot of study use spade, auger if we want to take deep soil, cylinder and spoon. And for sampling depth, it would be especially useful if we want to know how deep our microplastic penetrate into the vertically penetrate into the soil. So top with topsoil is normally the most the highly contaminated subsoil, and then in some sandy soil plastic can penetrate, and then it's really useful to take deep soil sample. And in agriculture field, normally we plow our soil down to 30 centimeter, so the recommendation would be to take at least 30 centimeter to depth of the soil. And of course, there has been also some studies studying down to 100 centimeter or 200 centimeter, if you look at the graph C here. But again, most of the study look at plastic in the topsoil. And the next question, which is also one of the most difficult one, is how much area or how much the volume of soil should we take to obtain representative sampling? And the answer is really depending on how contaminated your field or your soil is. And so I share here one study who do numerical simulation of plastic particle in the soil, and they have two scenarios. The first one is when the particle are really dispersed in the soil, and the second one here. The second one is this one where the particle tend to aggregate into the soil, and this is more the realistic scenario. And we see that for both case, the higher highly contaminated the soil is, the less soil amount, the less volume we need to take. And for field that is really less contaminated, we really need to take a higher amount of soil to be representative. And of course, in the case where soil are really aggregate into the clay, the plastic really aggregate into the soil, we need to take much higher amount of soil compared to when the plastic is highly dispersed, which is really less common. And also, it also depends on how many arrow which you want to resend. For example, if you only accept 5% of your sampling arrow, then of course you would need to take much higher amount. If you want to take like 50% arrow, then you can take smaller amount. So the higher amount of soil, the less arrow it is. And it also depends on the size of plastic we want to sample. For example, if we want to sample macroplastic, like larger than 5 millimeter, then the abundance of these plastic are less than smaller particle, for example, 1 micrometer or even nanoplastic, where the abundance is much higher. That means if we want to sample for large plastic, we might need to consider taking a larger area of consideration, whereas for smaller, where the abundance is high, we just need to take a small amount. And for my last experience, I expect a sort of concentration from the literature, and then I use this theoretical simulation to estimate how much of soil volume I would need to take. And that is soil volume. And again, the next thing is the number of sample. How many soil samples should we take? Also again depend on the concentration, how contaminated it is, and also what type of arrow would we want to expect. And one interesting thing that this study find is that instead of taking several, like a lot of small particles, maybe it would be more useful to take large volume of soil, but then less sample. And then, so for example, when we want to study the field in my PhD that is less contaminated, would expect like to be a control field, we need to take a really large volume, and then we only take 5 samples to obtain the enough representative volume. And for example, this picture here is a 50 centimeter multi by 50 centimeter quadrat, and then we homogenize within this quadrat, and then we took the soil sample to our lab. And of course, in a lot of field, agricultural field where we have plantation going on, it's really difficult to take large volume. But then I think we could take core, but then we have to take several core of sample and then use a composite to obtain a representative sampling. Yeah, so key message would be to take into in consideration into what sort of concentration, you would expect for plastic in the soil, what size you would expect, and also importantly, what is the limit of detection in your lab, how sensitive your method is. If it's really sensitive, maybe not a high amount of soil needed, but if it's not sensitive and there are sort of contamination from the lab, then you would need more to overcome these LOD and LOQ of your method. And so this is some picture of sampling microplastic in soil. Always before sampling, we do a quick on-site characterization to understand the humidity of the soil and the electric conductivity, and then we chose the soil. The site, we make the quadrat homogenize the soil, and it's important also to have field blank when you do sampling. This could be a wet filter next to the site. I was thinking it's also nice if we have some burnt, like some soil that we burn already, and then put it next to it and then bring it with our storage and go to the lab, so we can also have something similar with our soil. And then store it in 100% cotton bags. And also it's important to measure the bulk density of the soil that we're sampling, because we could also convert our data into volume or into mass when we know the bulk density. And after we have the sample that we collected from the field, we then bring them to the lab. What I normally do is also first do soil physical chemical characterization, so that we could know some more about the size distribution, how many clay, silt, sand the soil have, what is the organic carbon in the soil, pH and EC, because this will further affect the quantification of plastic, which I can discuss also later. And for microplastic extraction, after we have the soil, we normally dry. At the moment, there are 3 common methods for drying soil, like air drying, freeze drying, or oven drying. And oven drying is the most commonly used because we can dry a large amount of soil, and it saves time. And then we grind the soil, homogenize the soil, and then break the soil aggregate, and then sieve the soil, and then manual sorting. That is when we look for large plastic that can and put it under FTIR or microscope. And then the sieve size normally 1 millimeter or 2 millimeter, depending on what extraction method we want to use later. And after we have the soil sample that is ready for extraction, then we go to the extraction of microplastic. And again, I want to emphasize that this really depends on what type of plastic, what size of plastic you want to measure, and also what is the endpoint analytical techniques that we want to use. So this approach here is mainly for particle-based method, where we do density separation, filtration, and then removal of organic matter in the soil. But then for mass approach, we also have solvent extraction, which I not mention here today. So today I only focus on the most common approach. So for density separation, and second thing I want to emphasize is there is not yet any standard protocol for microplastic extraction on soil. In Europe, there is one effort to standardize the method now, but it's not finalized yet. So this really depending on what chemical and what equipment you have in the lab. For density separation, we have several setup. It could be this sediment separation unit, or you can use the separating funnel or freeze store of overflow setup, where you use a very, for all of this, we use high density of salt and then extract the plastic that is floating on the top. And for each of these techniques have this advantage and also disadvantage that we need to consider when we take it. And then after that, we take the supernatant and then filter it with different, again, filter pore size depending on our research interest, which size we want to measure the plastic. And then after that, we do chemical digestion, either using Phan, hydrogen peroxide, or enzymatic digestion. So this is just an overview of what chemicals are currently being used. The table 4, this is an overview of different salt that is used for plastic density separation. And we can clearly see that depending on the type of plastic that we want to measure, that we need to take the density, the appropriate density. For some really light plastic like LDPE, even molecule water or sodium chloride can work very well. But then for some plastic that is heavier like PVC or tire wear particle, we would need a higher density salt. And we like, for example, SPT, calcium chloride, zinc chloride. But those also have its advantage and disadvantage. For example, zinc chloride is really corrosive, and we also found in one of our study that it can be damaged to some of the plastic, for example, polyamide. And then SPT is a good one, but it's quite high cost salt. And for size fraction, most of the study now doing microplastic in soil look at very big fraction of plastic, like, for example, smaller than 5 millimeter or larger than 100 or 200 micrometer. And most less are looking to, like, for example, below 50 micron or below 25 micron. And for our previous experience, the smallest size that we go for microplastic is currently 6 micrometer or 25 micrometer. And for organic removal, the most commonly used is hydrogen peroxide and then Phan and enzymatic. Enzymatic is efficient, but it's time consuming and more costly. And Phan reagent is the really fast approach, which we favorable to use. And next is quality assurance and quality control, which is really important. So before, I already mentioned about sample collection, how the size, the volume, and the number of sample is really important in defining the error from the sample collection. And then storage is also something we need to consider, because it also can bring short of contamination or error to the analysis. And then in the lab, there are secondary contamination coming from our equipment, from our lab workspace. That's why it's really important to have procedure blank. So blank from the field, blank in the and also blank in our lab. And next one that define the reliability of our measurement is also how the sample recovery, that define how well our extraction, our quantification and sampling method is, mainly extraction and quantification. So with this, we normally spike into the soil sample with reference materials, and also in some case we can also spike in something that cause false positive. For soil is mainly organic matter. And then for reporting also is necessary to report limit of detection and limit of quantification of our method. So here is some measures that we normally do to reduce secondary contamination in our lab. Include breaking all our glassware, at high temperature for 4 or 6 hours, and then working under the laminar flow using 100% cotton lab coat, and importantly, filter all the reagent before use if we don't know how contaminated it is. And of course, it is most important to use procedure blank starting from the point we start the extraction and at the final stage where we measure our plastic samples. And with the procedure blank, we will be able to identify what is the limit of detection and limit of quantification. So limit of detection would be the average of our plastic concentration in the procedure blank plus 3.3 times of the standard deviation. And for limit of quantification, it will be the same except for this 10 times of the standard deviation. And all the reported concentrations should be subtracted to the average of the procedure blank. And recommendation would be to have 7 procedure blank for your method. And for some of the mass approach, we can also have limit of detection from our calibration curve. Which is using the slope of the calibration curve and the error from the calibration concentration. The next one is recovery test, which is really important also for microplastic analysis. So normally we should test our method with different type of plastic and different size range. And for the case of soil, because it's really heterogeneous, so it's also important to take into account the soil texture and soil organic content. So this is just an example of one of the study that I did, where we test different type of plastic and different size. We spike different size of plastic into the soil. We apply it through the whole extraction procedure that we have, and then we find the recovery. And we found that the recovery for plastic is different depending on the size. For example, larger size of plastic, which is the white column, is have much higher recovery than the smaller one, and is not really dependent on the soil type. But for smaller plastic, then it's really depend on the size. For example, on the type of soil. For example, LDPE, the gray box. For clay soil, we see a much lower recovery. This could be due to the loss of particle in the clay aggregate that we have during the density separation. And for biodegradable plastic, we also receive a lower recovery compared to conventional plastic, which is explainable because we might have the loss of the plastic due to the very strong, for example, Phan digestion that we use, or also the very corrosive zinc chloride salt that we use for density separation. And again, I mentioned that we can have really different extraction method depending on the endpoint of the analysis. For example, for microscope, is much more microscopy techniques is much simpler approach. But for pyrolysis, GCMS, Raman, or FTIR, we really need to clean our sample enough so that we will not obtain false positive results from the soil organic matter, and the procedure will be much more complicated. And also, depending on the techniques that we use, we also have different recovery. For example, for biodegradable plastic, we find that NMR was the most efficient method to extract biodegradable plastic. And then we also found for really small LDPE microplastic particles, fluorescence seem to be more efficient in detecting those compared to FTIR or Raman. So a lot of things, yeah. So that is my presentation. I just want to give some key messages. So at the moment, there are really a lot of methods available, and we can use different methods depending on our lab status. But what is important is that we need to report QA/QC for all of our analysis, including those that I mentioned before. and like recovery, LOD, LOQ, and also reproducibility of your method, how is the error range, and also need to address for positive and negative of the different method that we use. So I find some of these papers really helpful as a guidance on QA/QC that you could have a look. For example, in this one, they list different sort of criteria for reporting microplastic, like error progression, how the number of replica, limit of detection, and quantification. So when we do the study, we can refer back to this paper and check if we can tick all the box. Yeah, so that is my presentation.
Thank you, Nufa, for a very detailed presentation. As we all know, soil matrices are highly complex, so the presentation you given with such pictures, sampling and extraction methods that you use, and then leading to importance of quality assurance. In microplastic research, I'm sure that participants are now aware of your work and the work that we at InPlasTwin are also carrying out. And as we are a bit out of time, I would ask participants if they have some questions to Nufa, please post them in the chat. And according to the number of questions, we will decide if we are going now with the questions or we will leave them for the Q&A section.
Thank you, everyone.
And a moment in the chat, I don't see questions, so then I suggest that we move on and leave space to the participants to. So we have one question, sorry, from Erika. I think we have time to answer. What is the recommended temperature to dry soil?
Yes, so the recommended temperature would be like below 60 or below 40 for depending on the plastic that you analyze. For biodegradable plastic, then it should be, I think, below 60 degrees. And also if we want to analyze some plastic that really age in the environment, then it should be better to use really ambient temperature. So I would say 40 or 40. If higher than 60, it shouldn't be more than that.
Thank you, Nufa. We have one more question regarding the soil sampling, but I still don't see the question. Siebel Evans, would you like to write the question? Okay, I will leave this. Okay, catch up later. Okay, then we will leave this question for the Q&A section. And now I'm giving the floor to Dr. Fred Olav Libnau from Institute of Marine Research, who will give us some insights of microplastic research in sediments.
Hello, everybody. I'll try to share my presentation now. Hopefully you will see it.
Everything is perfect.
Good. First of all, thank you to the organizer for inviting me to hold this presentation. I call it the separation of microplastics from marine sediments, and I will focus on the method we are using at IMR. The institute is situated in Bergen, in a park at the north skirt of downtown Bergen. We have several buildings where the institute has a laboratory, and the sediment separation is taking part in the yellow buildings down at the harbor side. It's a building from 1819, so we can't do everything we want to do in the building because it's restricted. We have a more modern location where we have the instrumentation, so the samples are transferred from the seaside to the instrumentation lab during the procedure. Sediment sampling equipment. We have several different sampling equipment. Mostly used is the multicorer, especially in soft sediment, that is sediments that contains a lot of mud or clay. And going to coarser sediments, we have to apply a Phan grab. The samples can be taken out on board the ship immediately after sampling. If you're using a multicorer, then it's sliced into 1 to 2 centimeters thick slice and wrapped in aluminium foils. Nowadays, we do not do the slicing on board the ships. The pipe is sealed and the whole core is brought to land. We don't freeze the core on board the ship, but it's chilled. If you freeze the core, there will be some changes and disturbances within the core. And of course, when you're dealing with sampling in the Barents Sea, the crew has to wear PPE. We can't avoid that. Now talking about the density separation of microplastics from sediment, it's of course the act of separating low density particles from high density particles. And looking at the density of different particles, we see that the plastics has a wide range of densities, but also natural materials and coal or charcoals is distributed all over the place. And the density of the salt solution determines which particles end up in the upper or lower part of the solution. Sodium chloride is a cheap and also frequently used salt. The drawback is that the saturated solution of sodium chloride only extends to 1.2 gram per milliliter. We are using zinc chloride and calcium chloride. The equipment you use should allow mixing, weighting, separation, and collection, not necessarily all in the same equipment. It could be different equipment for the different phases. We are using a pipe for the density separation. We call it a Biota. It's a total height of 65 centimeters, so it's much too big to fit into a laminar airflow cabinet. It's essentially 2 parts. It's the separation chamber and a sedimentation chamber. The interesting particles is collected in the separation chamber. From the lab, we filter the tap water. We are using a lot of water, filtering it on 10 micron steel filters. We use a dust box to provide clean air. We have lowered the ceiling and the floor has been increased, so the total volume the dust box has to provide clean air for is minimized. And we are using 2 of these Biota each day. So when the Biota has been standing overnight, the valves, both the butterfly valve and the ball valve is closed. And before separating the 2 different chambers, you have to take out the solvent, which is below the butterfly valve. Otherwise, you will have a big leak into the lab equipment. And the top part is inverted and set into a filtration rack. And the filtration is first going through a 350 micrometer sieve to take the largest particles, and then through a 20 micrometer filter, steel filter. We wrap the steel filter, fold it, and double fold the open ends and secure it with a string. And when that is done, the particles inside the filter is protected from outside contamination. We do the chemical digestion on those bounded filters. It works down to approximately 20 micrometers. It doesn't work with 10 micrometer filters because the penetration of the solvents is not good enough. And especially when you are transferring the sample from one building to another, it's good to have the particles contained within a filter. Chemical digestion, we use several steps. We use 1 molar HCl for 30 to 60 minutes using a shaker board. Then the filter is flushed with tap water. Then we use a combination of urea and sodium hydroxides. We freeze it overnight and then thaw it until room temperature is reached, again using a shaker. And the final, it's an oxidation process where we use 30% hydrogen peroxide with some sodium hydroxide to speed up the reaction. And the reaction runs for 4 hours, again using a shaker board. The oxidation can also be neutral or on the acidic side, like the Phan, but we are using on the alkaline side. So this is an overview of the whole procedure as it was from the origin. Incoming sediment, some 100 to 200 gram wet weight, straight into a suspension of the salt solution, doing the density separation, sieving using a steel sieve. The particles on the steel sieve is transferred and investigated using a stereo microscope and ATR, FDR. The material on the 45 micron steel filter is run through a chemical digestion, and then we have 2 options, doing in a micro FDR or micro QCl-IR. And the results from the micro FDR are processed using simple, and the results from the micro QCl-IR are processed using machine learning models. And after that, we can run samples through, excuse me, releases GCMS and then reporting everything. During my 4 years at the Institute, we have done quite a lot of changes to the method. We started using zinc chloride and calcium chloride in combination. Now we are phasing out the calcium chloride. We start using wet sediment. We have now this autumn start using freeze drying. And I have to say, maybe we will return to using wet sediment again, because there is some problems with that. And if we can't solve it, then we have to return to using wet sediment. The benefit of using freeze drying, that is, it's very easy to make a homogeneous suspension. Without freeze drying, you have lumps, especially when you have a lot of mud or clay. And I use a lot of time to dissolve these lumps, because the lump is heavy, and if there is plastic inside it, it will just go to the bottom of the density separation column. We have changed from 45 micron filter to 20 micron filter. This spring, I separated some 91 sediment samples without problem using 20 micron filters and wet sediment. This autumn, I'm using freeze-drying sediment and separated some 21 samples where I have a clogging on the 20 microns filter in about 50% of the samples. And we also changed the chemical digestion using TO-urea, urea in combination, and avoided TO-urea. TO-urea is can be cancerous, and we have taken it out of the procedure. It's a very time-consuming process. It takes some 70 days if everything is perfect. Of course, the 7 first day is freeze drying, so at least 10 days if you start with wet sediment. And we avoid procedures extending into the late hours and weekends. That's important for me. So everything I do should be within normal working hours, or being able to leave in the freezer or whatever overnight so we can continue the day after. If one operator do the separation, he or she can manage to do 8 samples a week. If 2 shifts and support each other, we can manage 10 samples a week. And samples are made both sediment samples and method blanks. So it's not that many samples that we can run through the procedure during 1 week. This is some pictures from the 315 sieves, and it is tutti frutti, everything. Beautiful creatures we find in the samples. And this is when we have freeze-dried the samples, so every items that has a hollow inside, it's with the possibility of being filled with air, will float to the top, both plant and animal residue. And that is beautiful to look at, but not necessarily so when you think of what you got on your filter. When you have a lot of foraminifers in the sample and you freeze dry it, and you might end up with an overload. And this is an extreme case. I didn't do any acid treatment, and the suspension was transferred as quick as possibly to the Biota, and you end up with a lot of debris on the filter. And the debris will both include low density and high density material. And of course, when you get these kind of result, you have to do a second density separation. Of course, before you do that, you have to treat the sample with acid in order to dissolve the calcium carbonate. Unfortunately, there is also other shell or animal residue that is not made of calcium carbonate, but silicates and other stuff like chitin that acid treatment does not help for. That could be a problem. We have visual effects sometimes that both the urea treatment and the oxidation treatment works, but as we see, we don't every time see a color change on the solution. So the treatment works for some materials, but not for others. And that's how it is. We recycle the salt solution. As of today, we recycle some 96-97%, and that is necessary because every day we do the density separation, we use 5 kilos of zinc chloride, and we can't throw that away every day. So we need a centrifuge, and after the centrifugation, the salt solution is filtrated through a cellulose filter with a pore size of 6 microns and stored in PE bags, and prior to use it's filtered again, but then on a steel filter with a pore size of 10 microns. And that should be good enough when you are collecting particles. That should be bigger than 20 microns. If you want to go further down, the effort you have to put into the recovery is increasing tenfold. If you have ball valves involved, the wall valves should be dismantled and cleaned. There is cavity within the valves that is not easily cleaned without dismantling the valve. How much time I have? 3 minutes left. A few words on, you have to run blanks on regular intervals. You can set up control charts for both method blanks and air blanks. You can have the total number of microplastic particles, total number of natural particles to see how the air is contaminated over time. Or you can have different control charts for different plastic types. Remember to state the cutoff size when you do the counting and publish the results. This is from metal blanks and air blanks, and we see the difference in the number of particles when you have a cutoff of the ferret max for 45 microns and the ferret max for 20 microns. It's about a tenfold increase in the particle numbers. And I would also like to remind you that you should state the cutoff for the ferret mean or the minimum axis. This is how it turns out. If you have no cutoff, that is 1 micron, then in effect it's no cutoff on the ferret mean, and if you have a cutoff of 10 microns on the ferret mean. Why should you have some cutoff on the ferret mean? Because the number of particles with a ferret mean less than your filtration mask, it's not quantitative. It just say you how many of those particles you collected on that run, not how many of it was original in your sample. And you need those numbers in order to evaluate results over time, because here we have results from the Marano on microplastics in surface sediments. And it's very varying, and we can't without those numbers say if it's due to the reporting, the analytics, or the sediment sites and the actual number of particles in the sediments. Thank you for your attention.
Dear Fred, thank you very much for your presentation. I have to say that pointing out the adaptations that you have made during your research and working with sediments must be of high importance also to other colleagues that deal with sediments, so they can adapt and compare to their methods. I see a lot of hands congratulating on your presentation, and of course thank you for taking care about the time of your presentation. At the moment, we are supposed to have the coffee break, but if you allow, I saw one question in the chat, so maybe we can answer to that one, if you agree.
It's okay by me.
There was one question, but no. Okay, sorry. What are the filters used in this procedure made of?
When we are filtering the separated microplastic and organics and other stuff, we are using stainless steel filters.
Okay, I should.
We use stainless steel strings that is woven, and we use filters that is the same distance in both direction in the woven matrix. Okay. You can also have filters that has different dimension in the strings, but that could lead to problems getting the particles out of the filter again.
Thank you very much. I would use the opportunity just to say for the one who joined maybe later that this workshop is being recorded. So in case that you missed something during the presentations, you can see the recordings on our YouTube channel after this workshop. Now I invite you that we all take a short break until 11:30. So next 10 minutes, we will be back with our next two speakers on the second type of matrices that we will tackle during this workshop. Thank you for your participation so far.